All right, so let's go into making the protein. So this is a process called translation. So once again, we have initiation is the first step in translation. And what happens is the ribosome in the cytoplasm will find a mature mRNA and create a ribosome mRNA complex. So here we have the large ribosomal subunit and the small ribosomal subunit surrounding this mRNA, the mature mRNA. And what we see here is basically the ribosome scans the mRNA until it finds this AUG, which is the start codon. So AUG is always the beginning of translation, and it also encodes for a methionine amino acid. And so what this red rectangle here is actually a transfer RNA. So you can see it has the sequence that is complementary to the AUG sequence that comes and recognizes the AUG, and then the methionine comes in here. And so I just want to take a pause now because we haven't talked yet about codons. So this AUG, this trinucleotide codon, encodes for methionine. We have 20 different amino acids that are encoded by the 64 different possible codons. And so what you can see here is that there are many different combinations of these like trinucleotide codons that will still encode for the same amino acid. And this is what we call degenerate. So the genetic code is degenerate, and this is what we mean by that. Oops. Okay. Yeah. I'll get more into that a little bit later, into the different codons and amino acids. So we also have elongation as our next step. So the transfer RNAs, after bringing in the first methionine, which is the start codon for translation, transfer RNAs that have these codons complementary to our codons that I just showed you, brings amino acids to this ribosome mRNA complex, and they're added to this polypeptide chain. So here UGG encodes for a tryptophan, and then the next one UUC, you can see the transfer RNA coming in with a phenylalanine. And so this is how the polypeptide chain is created in the ribosome. And then finally, at the end, termination is signaled with a stop codon. So there are 3 different stop codons in our trinucleotide codon map, and they do not code for any amino acid. So while the start codon does encode for methionine, and you can also get other methionines along inside the polypeptide chain, the stop codons do not code for anything. It just signals, okay, this is the end of translation, the polypeptide chain is complete, we have our primary polypeptide sequence, and then the folding and everything else with the secondary and tertiary structure can occur after that. And so just as DNA and RNA and nucleic acids have their directionality where we list 5' to 3', so do proteins. So polypeptides have this N terminus, which is the beginning, and the C terminus at the end. So just for your future reference. All right, so let's get back to these codons. When we're looking at mutations, if we're looking at gene coding regions, we're kind of interested in being able to interpret what these mutations what sort of problems in the protein would these mutations cause. So I just want to go through a few examples to kind of give you guys like an idea of what we mean when we're talking about these different types of coding mutations. So let's first take a look at this UUA sequence. So the UUA codon encodes for a leucine amino acid. And so can anyone tell me what the sense strand sequence would be for this codon from the DNA? So does DNA have uracils? No. So then would the sense strand even include U's? Would the sense strand have an A, adenosine, adenine? The sense strand would? So remember, the template strand is the strand from which the mRNA is synthesized, or the RNA is synthesized, right? But the sense strand is actually the non-template strand, and it's the strand that you would look at if you were just interested in what your mRNA sequence would ultimately be, right? It's the reverse complement of the template strand, which means it's what the RNA will ultimately be. So what would the sense strand sequence of UUA be? It would just be TTA, right? All right, so let's say in that region of that gene, we have TTA, and the A gets mutated to a G. So what would the new codon be? Instead of TTA, it's now TTG. What would the new codon be? UUG, right? So if the A is mutated to a G, then the new codon would be UUG, and the amino acid that's coded by that codon is still leucine, right? And so if we were to have that mutation, we would call that a synonymous mutation because the amino acid that's coded does not change. All right, what if the TTA was mutated to a TTT? What would the new codon be? UUU, right? So what amino acid does that encode? Right, so we go from a UUA, if that A is mutated to a T on the sense strand of the DNA, then the new codon is UUU. That's a phenylalanine, and that's what we call a non-synonymous mutation. And it's more specifically, it's a missense mutation. So that means the amino acid that's encoded is switched, right? And this is actually a really important concept when it comes to the function of proteins. So a lot of times when people are trying to interpret the effects of a missense mutation, they'll look at what amino acid is getting switched to which amino acid is getting switched. If you have like two very small, similarly charged amino acids, it's possible the protein can still fold kind of regularly and function properly. But if you have like one small negatively charged amino acid getting switched to like a huge positively charged one, it's more likely that protein folding is going to be messed up, and then the function will be altered as well. So these missense mutations take a little bit more interpretation to figure out if they're actually, like how they might function. All right, so let's go back to our TTA sense strand. What if the mutation goes from TAA? What is the new codon? It's a stop codon, right? So if it goes from TTA to TAA, the new codon is UAA, right? And that's a stop codon. So we call that switch a nonsense mutation. So whenever you have a mutation that leads to not just an amino acid change, but an early stop, it's called a nonsense mutation. All right, and there's another type of mutation that can occur in the coding region, and those are indels. So we've discussed these types of mutations before. If they occur in the coding sequence, this can lead to a frame shift. So if you have an insertion or a deletion that's not in a multiple of 3, you are going to shift the entire open reading frame of your codon sequences and just like completely mess up all of the amino acids following that. This often results in an early stop. So it often results in nonsense mutations. But if it's a multiple of 3, let's say you have a deletion of 3 base pairs, what happens? Or you just lose an amino acid. Yeah, so kind of a missense. It's like one amino acid goes to no amino acid. So yeah, if it's a multiple of 3, it's a little bit better because your open reading frame will be maintained. But if not, then you'll likely result in a frame shift and then likely a nonsense mutation. All right, does anyone have questions about that?